Very good afternoon. My topic for presentation is immobilization of enzymes, methods and applications. What are the important parts of the immobilization of enzymes? First of all, we know enzyme immobilization. Enzyme immobilization is nothing but that is a process confining the enzyme molecules to a solid support over which substrate is passed and converted to products. So these are the important thing where we can insolubilize a particular enzyme is having more important in application of various processes. So what is an immobilized enzyme? That is nothing but where one whose movement in space has been restricted either completely or to a small limited region. So in the part of introduction, so many enzymes which are secreted by microorganisms are available on very large scale, and actually there is no effect on their cost if they are used only in a process. But there are many more enzymes are having very more value, and they have having very economically not affordable. So these enzymes we can reuse for the many processes. So here we can use immobilization techniques. So it involves the conversion of water-soluble enzyme protein into a particular solid support, that is a form of catalyst, by several methods. It is possible to immobilize microbial cells by similar technique. In the introduction part, these enzymes is attached to inert insoluble material such as sodium alginate. This can provide increase the resistance to changes in various physical parameters such as pH and temperature. So here insoluble material allows enzymes to be held in a place through the reaction, and here we cannot solubilize that enzymes easily in a solution. So here there is a separation of that product, it's an enzyme, from the solution easily. Okay. So in this process, an alternative enzyme immobilization is we can use as a whole cell immobilization. So why immobilization of enzyme is necessary? Because we can protect the enzyme from degradation or deactivation. Means what? Here we can use that enzyme for many reactions or many times. So here if we go for the reuse of that enzyme, of course we can lowering the total production of the cost of enzyme mediated reaction. So here this is important. Why? Because ability to stop the reaction rapidly by removing the enzyme from the reaction solution. We can enhance stability of that enzyme, and we can also improve the process control with the use of enzyme immobilization. So this is what easy separation enzyme from the product, and here we cannot contaminate the product with enzyme, or there is a less chance of contamination in products. So there are different carrier matrices for the enzyme immobilization. These are the inert, having physical strong and stable properties. They are cost effective, they should be regenerable, and they should not inhibit the product formation. So what are the different methods of the immobilization of enzymes? So there are 5 different techniques. Immobilizing enzymes placed in 2 types or 2 different groups. We can form here physical and chemical. In physical, adsorption, entrapment and encapsulation, and in chemical, covalent binding and cross-linking co-polymerization. So this is the example we can see in this picture. Enzymes already immobilized with a particular solid insoluble matrices or support. So we can use this enzyme as a reuse for many reactions. So green is nothing but enzyme, brown color is a solid or porous support. These are the mesh-like part is also porous polymetric matrices, and this line is nothing but semi-permeable membrane. So in case of physical adsorption, here this adsorption process where enzyme onto solid is probably by simplest way. How? Because here the bonding of this enzyme to solid, support or inert material is just because of a particular material. So here these are the alumina oxide or organic support. The energy, it's nothing, it's bond of low energy are involved, predominantly ionic interactions, hydrogen bonds, van der Waals forces. These enzymes are externally very important. They have limited particle size and where we can achieve their appreciable surface bonding. So there are further 4 procedures we can immobilize enzymes by adsorption by the interaction between enzymes and inert material by ionic interaction, hydrogen bond or van der Waals forces. So these are nothing but solid process. So here these enzymes immobilize on the carrier by allowing the solution containing enzyme where to contact the carrier without steering, that is static or stable process. Dynamic batch process where carrier is placed onto enzyme solution and mixed by steering or agitate continuously in a shaker, right? So in another way, reactor loading process in which carrier placed in particular reactor and here enzyme solution is transferred to reactor where there is environment is continuously agitating. So carrier and enzyme solution both are the moving and during this process enzymes molecule will adsorb. And finally in the physical adsorption process, the electrodeposition, in which process carrier is placed proximal to one of the electrode of an enzyme bath and current put on the another end of the carrier. So here there is a formation of what electric attraction between two different molecules and there is a formation of what deposition of the carrier and enzyme molecules. Another important part of the enzyme immobilization is what? Entrapment, in which process enzyme can be physically entrapped inside a matrix, that is nothing but a support. So these matrices are the solid soluble polymers such as polyacrylamide type of gels. These are naturally derived gels, example agar, gelatin, carrageenan, alginate, etc. So the form and nature of the matrix are vary. So here in this process, pore size of the matrix should be adjusted according to prevent the loss of the enzyme from the matrix, because there is a quite possibility of the excessive diffusion. So there are several methods of the entrapment: inclusion in gels, inclusion in fibers, inclusion in micro capsules. So you can see here enzymes are entrapped in a matrix, that is what inclusion in a gel, and entrapped in a droplet. So enzyme is in capsule. So you can see here this is a picture of enzyme molecules. These are what added or covered by a particular material, that is nothing but a polymer. We have entrapped these enzyme molecules in a support and we can see here the perfect insolubilization of this enzyme. In another way, enzymes we have encloses in a particular capsule or droplets. So this is nothing but in an encapsulation of the enzyme molecules. So what are the disadvantage of this entrapment? There is a quite possibility of enzyme leakage into solutions. There is a diffusional limitations and sometimes there may be a reduced enzyme activity and their stability, and here lack of micro environmental conditions. So this is very important. So these advantages should consider, and according to that we need to immobilize that enzyme onto particular support. Encapsulation is another way where we can enclose of enzyme molecule in a droplet of solution, and here this capsule in a semi-permeable membrane capsule. Of course this semi-permeable membrane capsule is made up of cellulose, nitrate and nylon. So this method of encapsulation is very cheap and simple, but these are depends on the stability of enzyme and catalyst is very effectively, because within a capsule the activity or catalytic activity of that enzyme should retain. This technique is restricted to the medical science field. In this method, large quantity of enzyme is immobilized, but only small substrate molecule is utilized because these molecules will only interact to the membrane of the droplet of that encapsule. So that's why there is a very less possibility to diffusion of that substrate molecule from the outside to the inside of the capsule. So covalent bonding or binding, it's another part of the enzyme immobilization. It's part or type of chemical binding. In this method, covalent bond is formed between chemical group of enzyme and chemical group on the surface of the carrier. So this bonding takes place between pH, ionic strength and other variable conditions. So these steps are important where we need to use a particular coupling agent. These coupling agents allowing to activation process and form attachment of particular enzyme over the inert molecule. So there are different types of carrier which are used in immobilizations, such as carbohydrate, proteins, amine bearing carriers, inorganic carriers, etc. So in covalent attachment, that can be directed to specific group, for example amine, hydroxyl, tyrosyl, which are on the surface of the enzyme. So hydroxyl and amino groups are main groups where we can adsorb these enzymes on a particular carrier, and these can form perfect bonds. So here sulfihydryl groups may also involve, but there is least involvement in the formation of bonding. So there are different methods of covalent bonding: diazonation, formation of peptide bond, group activation. So in diazonation, that is a bonding between amino group of support, that is nothing but amino-benzyl cellulose, amino-selenized group, porous glass and amino derivatives, and tyrosyl and historyl group of the enzyme. So here support and amino derivatives of the enzyme, they can form a diazonation reaction to form covalent bonding. So this can also involve with the use of peptide bond. It's carboxyl group and amino group. Group activation is just use of cyanogen bromide, which are to a support that containing glycol group, okay, especially cellulose, cypadax and cyparose, etc. We can also use polyfunctional reagents. So these are the bifunctional or multifunctional reagent. Predominantly glutaraldehyde is very good bonding agent that can form polymerization of the enzyme. So here glutaraldehyde form bonding between amino group of support and amino group of enzyme. The major problem of covalent bonding is that enzyme can be inactivated by bringing about the changes in the conformation. So this is not perfect process where we can inactivate the enzyme easily by forming bond between support and enzyme. So here there is a quite possibility so many changes or reaction changes occur in the active sites. So covalent binding is technically more complex, require variety of very expensive chemicals. Of course it is a time consuming, and here these immobilized enzyme preparation are stable and leaching is very minimal. So many enzymes immobilized by suitable group in the surface. These are the hydroxyl, amino, carboxyl and sulfihydroxyl group of the amino acid. So cross-linking and copolymerization. In this method, covalent bonding is possible between various molecules of the enzymes via functional agents such as glutaraldehyde. So these are the enzymes are important. We can use with the use of many coupling agents. We can form cross-linking, or we can make the polymerization of all enzyme molecules here.